My name is Yana Zorina. I am currently a senior research scientist at Memorial Sloan Kettering Cancer Center. So I was initially trained during my undergrad and graduate studies as a neuroscientist, which is the field that I really love. But one way or another, when you get into that field, you also get highly exposed to cell biology. I work in a cancer center where we take all sorts of different cells, treat them with large collections of compounds, and then look at so-called phenotypic assays, which in most cases involves looking at them via microscopy, using many fluorescent tags and stains to see cellular responses to different compounds and drugs. And that way, rather than the traditional approach where you use logic to think, okay, if this is my signaling pathway, then maybe I can target A or B here to get the response that I want. In high throughput screening, you essentially take a huge collection of chemicals, throw everything against the wall and see what sticks. Cells obviously consist of proteins, right? And you can look for your protein of interest to see where it is expressed and localized in the cell. For that, you use antibodies that can bind to those proteins. And then once you apply antibodies that go in and recognize your protein of interest, you can then stain those antibodies with fluorescent tags in all kinds of different colors. And then you get images of the cells that show you which protein of interest is localized where. We capture these images and then we upload them into software where we can quantify all sorts of different parameters such as shape, intensity, number of cells, cell survival, death, anything you can think of. And that's actually the part of the job that I love. I love image analysis and looking at pretty pictures and turning them into numbers essentially to gather data. I got interested in biology and neuroscience, which is the study of the brain, probably around 6th or 7th grade. And I was determined to go into something biology related, but at the time I was kind of ignorant of the fact that you can actually go into science. I thought, well, if I'm interested in biology, then medical school is the way to go. In high school, I actually participated in a research program where I worked in a lab, and then I continued working in a different lab during college where I majored in neuroscience. But it wasn't until probably my junior year of college that I realized, okay, I like biology. I went into pre-med as the default choice, but it doesn't mean that I have to go to medical school. I can actually go into biology and research. And I remember while I had this idea that I would go to medical school, and then I realized that I don't really want to, this will always stay with me. I went to a career advisor in my college, and I said, I don't think this is for me. It makes sense to go to medical school, but I don't think I want to. And she said, basically, there are 3 stages of science: researchers who discover the knowledge, teachers who spread the knowledge, and then doctors who apply it. And that each of those stages is equally important. During the first year and a half of graduate school, you do rotations where you try out different labs for 2 or 3 months each. And that's how you decide which one to go into for your training. And I came in, I said, okay, I'm going to go do all of my rotations in the Department of Neuroscience until somebody recommended looking into the Department of Pharmacology. And I was pleasantly surprised how much neuroscience was going on there as well. But it gave me an advantage of looking at the other side, being exposed to basic cell biology, signaling pathways that control different processes in the cells, and how those can be applied in essentially any field. So for example, right now I'm working in a cancer center, but during graduate school, I studied signaling pathways that would potentially lead to regeneration of nerve cells. A lot of the same genes are implicated in cancer. I have always done a lot of art, but I was telling myself that I was too busy with my work, with family, and so forth. But then in 2016, I just had this acute sense that art is really a part of me. And as I started thinking about what I would like to do and what my art should be about, I thought, well, why not join kind of the sides of my brain? And it's been a lot of fun. There is a method called confocal microscopy, which uses fluorescent channels, as I described before. And the point of it is that the microscope is able to focus on a super, super thin section, so you know exactly what is on at that level. If you take confocal microscopy images, what would happen if you turn them on their head and recreate them in 3D? So that's what I wanted to do, to kind of get a better idea of what whole cells or tissues may look like. And thinking of fluorescence microscopy, I immediately thought back to my experience as a teenager making beadwork and thought, well, beads are great analogies for pixels. In order to understand why science is important and that it actually makes sense, people outside of the so-called ivory tower of academia should be able to understand and interpret the science that goes on behind closed doors, essentially. But there is a greater movement now on science communication, so that people don't think that it's ridiculous what happens in a research lab. And I think that art provides a great channel for starting these conversations and making science a bit more digestible to the general public that does not deal with it every day. I really like image analysis. It requires a lot of attention to detail. It's really rewarding and almost counterintuitive when you can take pictures and turn them into numbers that actually mean something. And I really like looking at individual cellular responses and how very minor effects can actually have a great impact on a larger disease area. For example, something that I came across relatively recently is a technique called cell painting, where you apply 5 or 6 dyes in different fluorescent colors. And rather than looking for a specific response that you would be interested in, you look at the overall cell state. And based on that, you can make an educated guess of what is actually going on in the cell and what processes are worth looking into and researching further. And it's just such a fascinating way to really narrow down what may be happening in a whole organism and make it much more approachable and manageable, and then be able to translate it to whole disease areas. The past few years, and probably the next decade or so, a lot of new methods are coming out, exciting new methods, including spheroid cultures and organoid cultures, which rather than the traditional method of culturing individual cells on a flat surface, you can now actually create three-dimensional tiny organs that you can put into a dish of different types of tissues that you're interested in, and essentially almost have a tiny organism in a dish, which allows you to minimize both studies in animal models as well as even potentially the amount of work that needs to be in clinical trials. I think one of the things that really surprised me is how many common pathways there are in all sorts of different tissues. It almost doesn't matter what tissue, what type of disease you're working on. One way or another, you're going to come across the usual suspects. It's probably, you can probably count on the fingers of your hands how many central genes there are that are important for essentially everything. And if you studied them in one context, you will know where to start and how to begin working on them in a different, let's say, disease model. You know, it's mind-boggling in a way that you kind of feel at home when you see a familiar gene name, let's say, but then you wonder, well, how does it have completely different roles in different contexts? So, for example, a lot of the proteins that I worked on in regeneration of the central nervous system, a lot of them are oncogenes that are implicated in cancer. And while we were trying to activate them, oncologists are actually trying to suppress them. So there is this very fine-tuned balance in the organism, and everything is good in moderation, right? So you need to find how to fine-tune everything that is going on so that you have just enough of it, but not too much of a good thing that could throw the balance in the opposite direction. Given the fact that I'm working at a core facility, my week is typically sprinkled with a lot of meetings, typically with either new clients or reporting on the status of existing projects. And in between all of that, I spend time culturing cells in the lab. And then there's a lot of imaging and image analysis. And then the rest of the time is spent essentially crunching the numbers and seeing which compounds may give you a desired response, going back to the literature to see if it makes sense, if somebody has found something similar in your disease area, and putting together PowerPoint slides to present it to other people. Art was a hobby for me that I decided to take to the next level. I think it's really important to retain your interests and not let them fall by the wayside. It allows you to feel like while you may love science, that it does not completely consume you, that you don't have to identify as a scientist 100%. There are other multifaceted sides to you, and that's actually what makes you interesting to other people, including within the scientific community. I would say go in knowing what you're interested in, but at the same time, be open-minded to what you might stumble upon, because you never know what might pique your interest on another day. And again, it's important to realize that what you learn in one area may become unexpectedly relevant in another one, and that you never know how different types of knowledge may come in handy. But in general, I would say it is important to have a plan, to know what kind of milestones you need to reach in order to get to the target that you have set for yourself, but at the same time, be open-minded to what may come your way.